Welcome back to the competency protein and amino acid metabolism. This is Dr. Wilma presenting a video lecture on metabolism of tryptophan. I get blamed for why you're zoning cause I'm making serotonin and then some melatonin. All these puns have got you groaning. I'm in chocolate, I'm in turkey. Now you're really feeling sleepy. Who am I? Can you guess? Yes, it is tryptophan. At the end of this video lecture, you should be able to enumerate the specialized products obtained from tryptophan and its importance, describe the synthesis and importance of serotonin and melatonin, mention the biochemical basis of carcinoid syndrome. These are the specific learning objectives. The first amino acid to be identified as essential amino acid is tryptophan, which is indole ring containing amino acid. Chemically, it is alpha amino beta indole propionic acid, which is aromatic, as well as heterocyclic amino acid, which is neutral in nature, and it is both glucogenic as well as ketogenic amino acid. Tryptophan is metabolized by two pathways, Kynurenine pathway and serotonin pathway. In case of Kynurenine pathway, tryptophan gets converted into Alanine, which is glucogenic intermediate, and acetoacetate, which is ketogenic intermediate, and a Formyl group which enters into the 1 carbon pool and NAD+. And the second pathway is serotonin pathway, in which serotonin is obtained by tryptophan. This serotonin can be utilized for melatonin synthesis, in which serotonin undergoes acetylation and then methylation, so that melatonin is obtained from serotonin. Only 1% of tryptophan enters into the serotonin pathway to produce serotonin. The initial reaction of kynurenine pathway is oxidation of tryptophan to formyl kynurenine, which is catalyzed by the enzyme tryptophan oxygenase, also known as tryptophan pyrrolase. Formyl kynurenine is converted into kynurenine by removal of formyl group with the enzyme kynurenine formylase. Formyl group enters into 1 carbon pool, and the kynurenine is metabolized to 3-hydroxykynurenine in presence of kynurenine hydroxylase. In the next step, 3-hydroxykynurenine, which is hydroxylated in presence of kynurenine hydroxylase, is converted to 3-hydroxyanthranate and alanine in presence of the enzyme kynurenase. So kynurenase is redoxyl phosphate dependent enzyme, and the alanine which is obtained in this reaction is glucogenic intermediate. So the deficiency of vitamin B6 results in failure of conversion of 3-hydroxykynurenine to 3-hydroxyanthranic acid. Administration of isoniazid, which is anti-tuberculous drug, induces vitamin B6 deficiency and results in excretion of xanthorrhinic acid in urine. In the next step, the 3-hydroxyanthranate can undergo two types of reaction. So when it undergoes decarboxylation, it results in NAD+ and NADP+ synthesis. Or it can undergo series of reactions in which the ring is first opened by oxidation to form acroleil 3-aminofumarate, and further reactions by removal of ammonium ion and carbon dioxide leads to synthesis of alpha ketoadipate. Alpha ketoadipate through next few steps is finally converted into acetoacetyl-CoA, which is ketogenic intermediate. So in this kynurenine pathway, we obtain formyl group which enters into the 1 carbon pool, and alanine which is glucogenic intermediate, NAD plus and NADP plus, which are the coenzymes of niacin, and acetoacetyl-CoA, ketogenic intermediate. Serotonin is synthesized from tryptophan by neurons, pineal glands, and intestinal argentophin cells. Normally, only 1% of tryptophan is converted into serotonin. In the first reaction, tryptophan is hydroxylated to form 5-hydroxytryptophan in presence of the enzyme tryptophan hydroxylase. This reaction is very much similar to conversion of phenylalanine to tyrosine in presence of the enzyme phenylalanine hydroxylase. Here again we require tetrahydrobacterin so that it is converted to dihydrobacterin, and in presence of the enzyme dihydrobacterin reductase and NADPH, there is a regeneration of tetrahydrobacterin and a molecule of oxygen is utilized, and molecule of water is released. 5-hydroxytryptophan undergoes decarboxylation in the next step so that 5-hydroxytryptophan is converted to 5-hydroxytryptamine, which is nothing but serotonin. Let us discuss about the specialized products which are obtained from tryptophan. Like any other amino acid, tryptophan also used for protein synthesis. And from the Kynurenine pathway, we have seen that there are several products which are obtained. For example, Formyl group, which enters into the 1-carbon pool. Then we have Alanine, acetoacetyl-CoA, NAD+, and the second pathway, that is serotonin pathway, tryptophan is converted into serotonin. So serotonin is also a specialized product which is obtained from tryptophan. And serotonin further undergoes metabolism by acetylation and methylation to form melatonin. Here we have the Kynurenine pathway in which tryptophan is converted into formyl kynurenine, and there is a release of Formyl group which enters into the 1-carbon pool so that N-formyl kynurenine is converted into kynurenine. And there is a synthesis of alanine from kynurenine undergoing reaction to form 3-hydroxykynurenine, and 3-hydroxyanthranilate and alanine are formed. Alanine is glucogenic intermediate. 3-hydroxyanthranilate can undergo two types of reactions, but major pathway is to convert to acetoacetyl-CoA, which is ketogenic intermediate. Only 3% of 3-hydroxyanthranilate can get converted into NAD+ and NADP+. So these are the specialized products which are obtained from tryptophan by Kynurenine pathway. The 3-hydroxyanthranilic acid, which is obtained in the Kynurenine pathway, can either enter into formation of acetoacetyl-CoA or formation of NAD+ and NADP+. Only 3% of 3-hydroxyanthranilic acid undergoes several steps to form quinolinic acid, which in turn undergoes decarboxylation to form niacin or nicotinic acid. In presence of enzyme quinolinate phosphoribosyl transferase, the phosphoribosyl group of phosphoribosyl pyrophosphate reacts with niacin to form nicotinate mononucleotide, and inorganic pyrophosphate is released. In presence of ATP, the AMP is added to form desamido NAD+, which is aminated to form NAD+, using glutamine as a source of amino group. NADP+ is formed by phosphorylation of 2'-hydroxyl group of ribose of AMP moiety. So, in this way, 3-hydroxyanthranilic acid is converted into NAD+ and NADP+. Tryptophan undergoes degradation by the intestinal bacteria to result in the production of several indole compounds. These are mainly excreted in the feces as scatoxyl and in the urine indole acetic acid. So tryptophan undergoes deamination to form indole 3 pyruvate, then undergoes decarboxylation to form indole acetic acid, which is excreted in the urine. So the major urinary excretory products of tryptophan are 5-hydroxy-indole acetic acid from serotonin and indole acetic acid from tryptophan. Indican is the potassium salt of indoxyl sulfate, which is obtained by further reactions of indole acetate to form indoxyl indican, so that the normal excretion of indican in urine is 4 to 20 milligrams per day. Liver and intestinal bacteria can convert tryptophan to tryptamine and then to indole acetic acid. So the major urinary excretory product is indole acetic acid and 5-hydroxy-indole acetic acid from serotonin. The other specialized product which is obtained from tryptophan is serotonin. Serotonin is synthesized from tryptophan by serotonin pathway with the help of the enzymes tryptophan hydroxylase and decarboxylase. So tryptophan hydroxylase, in presence of a molecule of oxygen and tetrahydrobiopterin, converts tryptophan to 5-hydroxytryptophan. 5-hydroxytryptophan is converted into 5-hydroxytryptamine, which is nothing but serotonin, in presence of the enzyme decarboxylase and pyridoxal phosphate. Serotonin is synthesized in the brain, platelets and argentaphin cells of the abdominal cavity. It's an excitatory neurotransmitter in the brain. It stimulates cerebral activity. It has got a major role in behavioral patterns, also the regulation of body temperature and blood pressure. It's a powerful vasoconstrictor. It increases the gastrointestinal motility and it acts as a precursor for melatonin synthesis in the pineal gland. In case of depressive psychosis, there is low level of serotonins. 5-hydroxy-indole acetic acid is also a major excretory product which is obtained from the degradation of serotonin in presence of the enzyme monoamine oxidase. Normally, in an healthy adult, only 1% of tryptophan is converted to serotonin. But in a condition where there is an uncontrolled growth of argentaphin cells of gastrointestinal tract, it leads to a condition known as carcinoid syndrome or argentaphinoma, where there is 60% of tryptophan getting converted into serotonin. Therefore, niacin deficiency or pellagra-like symptoms may also be seen in carcinoid syndrome. The diagnosis of the condition depends mainly on the detection of increased excretion of 5-hydroxy-indole acetic acid in urine, because serotonin, when it undergoes degradation in presence of the enzyme monoamine oxidase, it gets converted into 5-hydroxy-indole acetic acid, which is excreted in the urine. The clinical manifestations of carcinoid syndrome are flushing, abdominal cramps, bronchoconstriction, and fluctuating hypertension, sweating, and pellagra-like symptoms. Pellagra-like symptoms appear in case of carcinoid syndrome due to increased production of serotonin. Hence, there is decrease in the synthesis of niacin, which leads to the clinical manifestations such as dermatitis, dementia, and diarrhea. There is an interesting fact about the relationship between serotonin and the protein and the carbohydrate-rich diet. When you consume protein-rich diet, all amino acids are available in the blood, so that there is an overcrowding of amino acids to enter into the brain cells, or a traffic jam for the amino acid transport system to enter into the brain cells. Tryptophan, being a bulkiest amino acid, it is taken up very slowly. On the other hand, if you consume a carbohydrate-rich diet, insulin secretion is increased and it reduces the amino acid concentration in the blood. So, tryptophan, even being a bulkiest amino acid, it easily enters into the brain because there is no traffic jam now. So that the tryptophan gets converted to serotonin and it induces the sleep. So dear students, now you decide during the examination, would you like to have a protein-rich diet or a carbohydrate-rich diet? The last specialized product of tryptophan which we are going to discuss is melatonin. Melatonin is synthesized within the pineal gland from serotonin. Serotonin, which is obtained from tryptophan, undergoes acetylation in presence of the enzyme acetyltransferase, and acetyl-CoA is required for this reaction. So N-acetyl group is condensed with serotonin to form N-acetylserotonin in presence of the enzyme methyltransferase, and SAM donates the methyl group to N-acetylserotonin to form N-acetyl-5-methylserotonin, which is nothing but melatonin. Its synthesis is regulated by light-dark cycle and blood levels of melatonin rises at night. It not only acts as a hormone but also local regulatory molecule of various tissues. It is involved in sleep-wake cycle and regulates the reproductive functions and also acts as inhibitor of melanocyte stimulating hormone and ACTH. Do you know which hormone is involved in the process of circadian rhythm? It is the melatonin which is involved in the process of circadian rhythm. The amount of melatonin in our bodies starts increasing in the evening and peaks in the middle of the night, letting us know it is time to sleep. It then decreases by morning, allowing us to wake up refreshed. This process of circadian rhythm starts in the retina. When the retina is exposed to light, a signal is relayed from the retina to an area of the brain called as suprachiasmatic nucleus, which plays a role in making us feel sleepy or wide awake. The suprachiasmatic nucleus sends signals to the other parts of the brain that control hormones and body temperature. Then signals travel from the brain down the spinal cord and back up to the pineal gland where the synthesis of melatonin takes place. During the day, such signals prevent the pineal gland from producing melatonin, but when it is dark outside, these signals are not activated and the pineal gland is able to produce melatonin. In other words, exposure to light prevents melatonin release, which keeps us awake, and lack of exposure to light causes melatonin release, which tells us to go to sleep. Thank you all for watching this video lecture.